Welcome to Ask the Expert. It's a brief, informative, lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. If you have any questions for our guest, Jessica Kenison, today, please feel free to enter them in the chat or raise your hand at the end of the presentation. And as I just mentioned, today is our guest. We have our guest, Jessica Kenison, PhD, coming to us from Boston. I'm going to give her a little bit of her bio because it's just pretty phenomenal. She received her BA in neuroscience from Boston University, graduating cum laude. She worked as a technical research assistant in the lab of Francisco Quintana at Amnorami Center for Neurological Diseases at the Brigham and Women's Hospital in Boston for 3 years before she enrolled in this PIBS program at BUSM. And as a PhD candidate in the laboratory of Dr. David Scher, Jessica was investigating the role of the aryl hydrocarbon receptor in autoimmune disease, tumor immunosuppression. We'll hear more about this in a minute, but this is pretty significant. By the time of her graduation, she published 14 peer-reviewed papers and 13 abstracts, and also was a recipient of the New England Immunology Conference Young Investigator Award in 2019. It's pretty amazing. She's also a scientist at Adtolerix, which Pfizer has exercised an option to exclusively license an immune tolerance therapy from this company's T1D program, which is pretty exciting for her too. So I'm going to turn it over to Jessica. Can you give us a little thumbnail sketch of your sort of, where you are right now and what excites you most about the research you're working on, and possibly even throw in your newest paper if you're up for it. Welcome and thanks for talking to us.
Thanks so much. Yeah, I'm really excited to get the opportunity to talk to you guys. So, I mean, you gave a little brief overview there of my background. I'm currently working with Antolerix. I'm actually also working as a postdoctoral fellow at Brigham and Women's Hospital with Dr. Francisco Quintana. And so we're doing a lot of really exciting research these days. In particular at Antolerix, I'm going to tell you guys about our work focused on antigen-specific immune tolerance for the treatment of a wide array of autoimmune diseases. So not just type 1 diabetes specifically, but also how we think that this platform can really fit into a number of different diseases, which are all common in the way that autoimmune diseases really have improper activation of the immune system. Like you mentioned in my PhD, I also was working on tumor immunology. And really what was fun was that my PhD was this intersection of both autoimmune diseases and tumor immunology, because I was studying this particular receptor, the aryl hydrocarbon receptor, which you guys are going to hear about because it's the receptor that we focus on at Antolerix. And what's so great about it is that it sits at this balancing point in your immune system, and depending on which way you target it, so to suppress it in the case of cancer or to activate it in the case of autoimmune diseases, you can actually treat the different diseases, which present very differently, but in actuality have a lot of similarities in the way, at least, that the immune system is functioning. So that's pretty cool. We had a recent paper come out in PNAS just this past year, looking at the aryl hydrocarbon receptor in tumor immunology, and last year in PNAS, looking at AHR in autoimmunity and specifically multiple sclerosis over there. So a lot of really exciting work looking at this receptor, but also more broadly at the idea of really restoring the immune system to how it should be functioning in order to treat diseases, which I think is super cool.
Yeah, agreed. I think, do you want to dive a little deeper into, this from the current PNAS paper, basically, you've got a nanoliposome-based platform for the induction of antigen-specific tolerance via using this modulation of signaling by the aryl hydrocarbon receptor. Do you want to talk a little bit about that?
Absolutely, and I've got some slides on it, but liposome technology is really hot right now, given that all of these mRNA vaccines are delivered by liposomes, which is really exciting for all of us that were in the field and felt like it wasn't quite being talked about enough. And of course, that's like a totally different, well, not totally different, but it's a different application. But you can really see how liposome technology and nanoparticle technology can be adapted to a variety of different treatments. And I think it's really an excellent delivery mechanism. It's incredibly safe, which was one of the reasons that it was able to roll out so quickly with the vaccines. And one of the reasons that we think that it's such a great application or modality for the use of autoimmune diseases, where you're imagining potentially treating people long term. And so the focus on safety, I think, is really critical in the world of autoimmunity, in a way that, sometimes with cancer, other types of places, you can get away with having maybe some therapeutics that are a little harsher, but with nanoliposomes, they've got a really strong safety profile. You're able to target them really specifically, and it's really exciting.
Yeah.
Cool stuff.
No, this is really interesting. And I know, like, you guys, one thing that intolerance is really kind of, I guess I hope you'll show a slide where you have the, sort of the nano formulation diagram, because it really shows how you constructed such a really, a really interesting nanoparticle, really, that, it has, that has a lot of parts. I mean, it's not just maybe some people are imagining this sort of like, liposome that's just sort of floating in there, but this is a really engineered liposome, I guess I would say.
It is. Yeah, we want to make the most out of it. So how about if I jump into my slides and then?
Yeah, go for it. You're ready.
But feel free, absolutely, to ask questions throughout. I think that makes it more fun, at least for me.
Yeah, please do, everyone.
All right. Can you see my slides?
I can.
Perfect.
Pretty.
All right. Thanks. Yeah, we like dendritic cells, and they happen to be really beautiful. So Antolerix, we get our name from antigen tolerance therapeutics. It's a bit of a strange name, but really the idea here is that we are generating and engineering these nanomedicines for the treatment of autoimmunity and potentially other diseases as well. But really the idea here is that a lot of autoimmune diseases result from the improper activation of the immune system, right? So your body is improperly recognizing these antigens from the pancreas or from other tissues in other autoimmune diseases. And what's happening is that you have this induced adaptive immune response, where these T cells are infiltrating the tissue. They're causing a lot of destructive damage. And the idea here is that you already have pathways and mechanisms in place in your body existing already, which are meant to shut down these pathways. And that's what we kind of consider tolerance, right? Is the idea of teaching these T cells and adaptive immune cells that the antigens that they're being exposed to or that they're looking at are not anything to be bothered with, right? So this is a really nice diagram here. It's a very dendritic cell focused diagram, but that's kind of how we like to think about it. Antolerix is you have these dendritic cells which interact with a variety of T cells, but also B cells and other immune cells. And what's happening in autoimmunity is that you have these activation of all of these CD8 positive T cells or Th1, Th2, CD4 positive T cells. And they're recognizing these self-antigens and putting out all of these cytokines and really negative responses that, when they're infiltrating the tissue, can be really destructive. And dendritic cells are constantly in communication with these T cells. And depending on the signals that dendritic cells are putting out is really how a T cell is programmed and how it's responding. And so what you can do in order to induce tolerance is to induce this more regulatory phenotype in dendritic cells, which then go and talk to DCs or talk to T cells, and through the use of these immunosuppressive cytokines and tolerogenic receptors and things expressed on the surface, in order to convert these T cells or induce T cells into a more regulatory phenotype. So you might be shutting down existing CD8 positive T cells through things like anergy, or you might be inducing regulatory T cells, both conventional FOXP3 positive T cells, as well as IL-10 positive TR1 cells, which are highly immunosuppressive. And so the way that we do that at Antolerix is actually through this endogenous pathway called the aryl hydrocarbon receptor. And there's been a lot of work done on this particular receptor. Actually, fairly recently, it's really only been around or known for the last 10, 12 years in the context of immunity. It's actually known longer past back then. But really, what's interesting about the AHR is that it can be thought of sort of similarly to a steroid receptor. It's a receptor that, when inactive, sits in the cytoplasm, but it's activated by a wide array of ligands. And these ligands, when they bind to the AHR, activate it, and it translocates into the nucleus. And once there, it acts as a transcription factor. And it affects a whole host of different genes, that can change the morphology of the cell and change a number of different things. And in particular, it can have effects in dendritic cells, but it can also have specific effects in T cells themselves. The pathway that we tend to focus on at Antolerix, because our nanoliposomes are targeted to DCs, is that activation of the aryl hydrocarbon receptor in dendritic cells themselves actually turns on this tolerogenic pathway, particularly through the upregulation of IDO1 and the kynurenine pathway. And through this mechanism can actually suppress effector T cells and induce regulatory T cells. In particular, it induces FOXP3 as well as IL-10 positive TR1 cells, and it suppresses Th1 and Th17 cells. And as I mentioned, we can actually have direct effects on AHR within the T cells themselves. And so activation of the AHR pathway in T cells can change their phenotype and change their differentiation states more towards regulatory T cells and away from T effector cells. Although I will emphasize that that's actually ligand specific. And so there's a whole host of different AHR ligands which are known. It's actually quite a promiscuous receptor. They can be found in everything from your diet, things like broccoli and a lot of tryptophan metabolites are AHR activators, as well as different ligands being produced through your microbiota and actually through the gut microbiome, which is a really fascinating area of research that we're doing a lot of work on now too. And
it's fascinating. I just wanted to ask you quickly, Jessica, this is curious over here, where the proteasome is involved in the degradation of AHR?
Yes, to a certain extent, yep.
Okay, and so, that's really curious. And then the lipid, sort of the lipid piece is released and then it's degraded.
Yeah.
Is that ongoing, or is this kind of, is that very regulated?
It is definitely regulated. And actually there's this whole kind of AHR pathway that acts on itself. So it can both, it's an interesting self-regulation loop that AHR does, because actually the activation of AHR leads to the production of, believe it or not, more AHR ligands. So you can have this buildup and activation of the AHR pathway, but at the same time, it also can activate things which induce proteasome activity and induce the degradation of the AHR. So it's kind of this interesting self-regulatory loop. And actually one of the major genes that it does is the AHR repressor, which actually competes with AHR ligands in order to shut down the AHR. So it's got a really interesting self-regulatory feedback loop within the AHR itself, which is cool.
That is very cool.
Yeah.
Okay, sorry to interrupt. I just was sort of curious about that piece.
Yeah, no problem. And so the ligand actually that we focus on at Antolerix is one called ITE. It's got a really long name, so I don't like bothering. But basically, it's an endogenous ligand that was originally isolated from the lung. And the thing that we like about it, on top of the fact that it's endogenous and already exists in your body, it's incredibly potent. It's one of the most potent ligands, at least in our hands. And it has a very strong safety profile. It's actually really, really quickly metabolized into inactive versions, and it works really, really well at inducing this tolerogenic phenotype in DCs and inducing regulatory T cells. Because like I mentioned, actually different AHR ligands can have sort of different effects. Based off of, it's pretty contradictory, but it's believed that it's possibly through the way that ligands bind with the AHR, and they can change the conformation in order to induce different downstream effects and with different transcriptional factor partners and things like that.
That's cool.
Yeah,
and picture I was hoping for.
That's the picture you were hoping for, yeah. So the way that we choose to deliver our AHR ligand is actually through nanoliposomes, which are really cool for a number of reasons. But the reason that they're so perfect for us is that our AHR ligand, ITE, is hydrophobic. And so we're able to embed it in the lipid bilayer of these nanoliposomes, while at the same time we have this nice hydrophilic core where we can put anything water soluble. And so what we choose to do is to load these T or B cell antigens, and usually peptides, or we can even do whole proteins. And so that you can co-deliver to the same cell every time, you know that you're delivering both the AHR agonist in order to induce the tolerogenic phenotype, but also the disease-specific antigen, which you can trade out depending on which disease you're studying. And that way you know that the signal that you're inducing is actually very specific to that disease. and hopefully limiting off-target effects, but also really generating these regulatory T cells that then hone to the site of inflammation specifically and work in a really highly specific manner. And basically what's nice about nanoliposomes also is that just based off of their size and charge characteristics alone, we're able to target them to antigen presenting cells like dendritic cells, macrophages. And we're able to see really, really efficient uptake both in vitro as well as in vivo of these nanoliposomes. They're awesome in terms too of all the different tools that you can use, right? You can label these liposomes and track where they're going, what cells are uptaking them. And they work really, really well at delivering our HR ligand at the same time as delivering our antigen of interest. And so, as I mentioned, these particles can be used to activate the HR and induce a tolerogenic profile. This is just some different gene transcription or gene expression data that we've generated, looking in mouse dendritic cells, where if we incubate with our nanoliposomes, we see strong upregulation of the IDO pathway, which is immunosuppressive, as well as IL-10, TGF-beta, and suppression of pro-inflammatory cytokines like IL-6 and TNF-alpha. And this is the same regardless of whether you have the ITE, the HR agonist alone within the particle, or whether you're also co-delivering a disease-specific antigen. So you're able to load up both of them efficiently.
Wow.
And in addition, you're able to still be able to present antigen very efficiently. So these liposomes not only go to the DCs, but they then present in this case, what we were doing was using cells from a 2D2 transgenic mouse. So these are mice that have all of their TCRs, or all of their T cells express a TCR specific for a particular peptide called MOG. And if you take these T cells and stimulate them with our nanoliposomes, if we just have MOG in the nanoliposomes, they proliferate really, really well, which means that our dendritic cells are not only taking up the liposomes, they're presenting the antigen and able to stimulate the T cells. But if we have our ITE in there, we actually don't see this proliferation because we believe of this tolerogenic phenotype that's being induced and the activation instead of regulatory phenotypes, thereby suppressing the proliferations. And this is the same whether we look in both mouse DCs or human DCs. We're able to really strongly activate the HR, induce immunosuppressive phenotypes, and suppress pro-inflammatory cytokines.
It's nice you have both mouse and human data. Very nice.
Yeah. It's important. I think we need translation. That's what we're all about for sure. And I'm going to speed through this a little bit, but basically what it is that we've done a lot of work looking at the pathways behind how all of this operates. And really what it is that our nanoparticles go in, they activate the aryl hydrocarbon receptor, and through a couple of different mechanisms, it's actually not just one, but a couple of different mechanisms, including the SOX2, which inhibits NF-kappa B, and thereby shutting down pathogenic T cells, or through CD39, an immunosuppressive IDO, or actually through some really interesting work that I can't talk much about, but that's coming out shortly out of Francisco Quintana's lab at the Brigham, where they're actually showing that AHR is remodeling the metabolism of the cells in order to, again, induce regulatory T cells. And what we're using it for really arrest autoimmunity. And in what you guys have all probably been waiting for, in our mouse models of type 1 diabetes, so if we were to look at the non-obese diabetic mouse, which is a spontaneous model of type 1 diabetes, treating with our nanoparticles, which are loaded with ITE, or AHR agonist, as well as either whole protein proinsulin, or we've also been able to show it with an insulin peptide. We're able to suppress the incidence of diabetes. We're able to suppress overall blood glucose levels in these animals. And we're able to do that actually by beginning to treat fairly late on. So we're, even if we don't start treating the mice until 10 weeks or even 12 weeks, when you already have damage to the pancreas at that point, we're still able to suppress disease. And we can do this actually through subcutaneous administrations or intravenous administrations, which is really positive when thinking about how we're going to translate this to humans, because something like a subcutaneous injection is going to be really preferable from the patient standpoint compared to having to do intravenous infusions. And mechanistically, this is happening through a decrease in pro-inflammatory cytokines and an upregulation of regulatory T cells, as well as if we were to actually take, this is some really exciting work that we did in the paper where we took T1D patients and we isolated dendritic cells. And even when we put in nanoparticles in these dendritic cells from human patients, we're able to suppress responses to autoantigens, in this case, GAD, which is one of the really common autoantigens in type 1 diabetes.
Yeah, that's really interesting. You know, because everyone has some level of of GAD in the body, yep. Right? GAD65, and they have some sort of like, they're tolerant to it. But so why is this GAD65 creating intolerance? I mean, this system and you've addressed it, so that's really great. But I mean, this is like a modular system. You could go one by one with each one of the potential auto antibodies and really just try to click away at them.
Exactly. So in type 1 diabetes, it's actually, it's really good one because I feel like the autoantigens are really well known and well characterized. That's not true in all autoimmune diseases, but in type 1 diabetes, it is thought there's a pretty solid progression at this point, right? If you have one or more or two or more autoantibodies to these known antigens, right, then you're going to develop diabetes if you have those early on. And that's known. And so you can actually think about going in with these patients that we know have these responses to these specific antigens. And if we could begin treating them earlier on, we think that we'd hopefully really not only be able to treat disease, but maybe even prevent it if we were to go into these patients that are expressing autoantibodies, but maybe not actually having full-blown disease yet. That would be ideal.
Yeah, it's almost like you would train it, right? You're re-educating, sort of
re-educating
the APCs one at a time, one at a time. I mean, how many would you imagine that you would, load up these particles with one autoantibody or antigen at a time, or would you do several? I mean, is there any idea how many you can add and have good outcomes?
Yeah, so we've been able to see that we can co-load multiple antigens, but obviously the simpler, the better. I mean, we're going in already to the FDA with like a really complicated particle. So on the one hand, the simpler, the better. So in type 1 diabetes in particular, we're thinking about using the insulin antibody or the insulin antigen, because that one's, generally the most common across all of the patients. However, it is advantageous for sure to think about going in with multiple autoantigens, because then you can,
maybe over time, like a time sequence of re-education.
Exactly. And I didn't have time to show it here, but it's actually in our most recent PNAS paper. One of the big strengths of Antolerix actually was that we were able to demonstrate that we can get tolerance not only to the peptide or protein that you're targeting, but actually we can induce bystander tolerance. And so we can get it against related antigens. So not ones that are, just any old random antigen, but what's happening is that these T cells are trafficking to the sites of inflammation and attack. So in type 1 diabetes, they're going to the pancreas, or in multiple sclerosis, they're going to the brain, and they're acting on the cells in that local area. And so what you can do is actually get this idea of bystander tolerance, where it's not just the antigen that you're putting in, but also the related ones in the disease. And we think that that provides us with a certain level of protection too, because maybe you don't have to get the exact antigen that's being targeted. Maybe as long as you're coming in with one of, say 3 antigens, maybe that'd be enough.
Yeah. So. And you can imagine that this would be a really interesting way to like an adjunct therapy to add to islet implantation, right? Because that's one of the biggest problems is the rejection of the islet, the implanted islet. So absolutely, because coming in with this, it seems very helpful, to say the least.
That's exactly the idea, because they're kind of two separate ideas of treating the disease, but you definitely would want to think about the ways to overlap them, because while we are trying to stop the cause of the damage, we don't necessarily have any evidence that we'd be able to restore pancreatic beta cell function of those that have already been destroyed, right? Mm-hmm. And so if we were to go in and co with another therapy that maybe does target and restore those beta cells, then that would be potentially very beneficial.
Yeah, there's many out there that are trying to, resurrect the beta cells also. I mean, Anil Bhushan's got his whole senescence and Peter Thompson, the senescence approach, where
Absolutely.
they
But
try
then
to resurrect them. And then, it definitely seems like it's going to be, it's not going to be a one and done. It's going to be a
right.
multi-therapy for this disease.
Right, because their problem is even if they're restoring the beta cell function and regenerating those cells, you're still having the original problem, right? The immune system is still targeting those cells. And so how you get around that, I think that's why we're going to need a multi-pronged approach.
Right, totally true. Who has other questions? Anyone in the audience? Silence. I'm surprised because there are so many, or even put something in the chat if you don't want to raise your hand. There's so many. What else? Have you come to the end of your slides? I'm sorry. We've just been, it's just really exciting research.
Oh, it's great. I much prefer a question answer type. I actually wanted to switch gears just a little bit just because I'm so excited about the work that we've been doing in multiple sclerosis. And like we were talking about, the fact is that this type of approach can really be used to target multiple autoimmune diseases. And what's nice is that we've been able to learn from each different autoimmune disease. And so I just wanted to really briefly touch on multiple sclerosis because this kind of highlights some of the different modalities and ways that we're able to treat, which can be absolutely applied to type 1 diabetes as well. And so this here is showing it's the EAE mouse model of disease. So this is where we induce disease in C57BL6J mice. And they specifically are targeted against this myelin peptide. And they, over time, develop paralysis. And we're able to clinically score this paralysis. And what we've been able to demonstrate was actually that treating with our nanoliposomes loaded with ITE and antigen in this case, which is MOG, we can treat them intravenously, we can treat them subcutaneously, we can treat them once they already have ongoing disease and actually reverse that ongoing disease. And in all instances, it's really driven by the upregulation of antigen-specific cells. So we're able to go in with tetramers specifically against MOG and show that we're generating these or inducing these FOXP3 positive and IL-10 positive antigen-specific CD4 positive T cells and really suppressing very specifically the pathogenic T cells specific against this antigen, which is cool because again, we really want to focus on antigen-specific induced responses.
Yeah,
and
I mean, it adds more to, it just adds, it adds more to the story really about what these particles can do. Exactly.
And this is like my last data slide, but I think it's just, it's too cool not to.
It is very cool.
That in multiple sclerosis, I'm not sure how familiar people are, but there's two different forms of multiple sclerosis. You have your relapsing remitting form, and then you have a chronic progressive form. And what's great is that these days we've been able to come out with a lot of very strong therapeutics that are very effective in relapsing remitting multiple sclerosis, and that's great. But unfortunately, a high subset of patients still develop chronic progressive multiple sclerosis or secondary progressive multiple sclerosis, for which there are very, very few treatments. And it's really quite a challenge. And we think that that's partially because mechanistically, things are happening differently in secondary progressive MS compared to relapsing remitting. But what we've been able to show is in a model of secondary progressive MS, where you have this initial acute phase of disease followed by this more chronic phase, if we begin treating with our nanoliposomes only in the chronic phase, so we were treating these mice once a week subcutaneously, starting from about day 35, we're actually able to reverse the disease that was ongoing and totally prevent this chronic progressive disease. And we showed in the paper that this was really mediated by this induction of IL-10 positive TR1 cells, which migrated into the brain and spinal cord of these mice, and then acted on astrocytes, microglia, and monocytes in the CNS in order to induce this regulatory phenotype and suppress pro-inflammatory innate immune cells, which I think is really critical. It's not just the adaptive cells, but actually through these TR1 cells, we're able to talk to innate cells as well and really make a difference. And I think that's what's going to hopefully be really exciting in secondary progressive MS, as well as, we showed strong data in relapsing remitting also.
Yeah, no, that is really powerful. That's fantastic.
Yeah, so that's my summary is that nanoliposomes induce tolerogenic DCs. They expand antigen-specific FOXP3 and IL-10 positive Tregs. They also suppress pathogenic T, and I didn't get to show the data here, but we can actually suppress pathogenic B cells as well. And these IL-10 positive TR1 cells suppress inflammation, which are driven by tissue resident cells. And so what's really exciting is that this is a platform. And we think that we have applications in a wide array of autoimmune diseases. Our lead is definitely type 1 diabetes, followed really closely by multiple sclerosis, but we're strongly interested in rheumatoid arthritis, things like food allergy, right, where your body is targeting peanuts. We think that we could treat as well, along with a bunch of different diseases. But I'm happy to take questions further.
Okay. Yeah. Let me see if I can go to, is there any, but let's see, here's in the chat. Thank you so much. I'm in awe of this, says somebody. Great talk. Thanks a lot. People are kind of, really enjoying this.
I should shout out, by the way, Sarah is here right now. Sarah was at our company. She has since moved on to do brilliant, wonderful things, and I can't wait to hear what she's up to these days. But thanks for tuning in.
Sarah, what are you doing? you can
put you on the spot.
Yeah. Please weigh in if you'd like to. I just, I mean, we're almost at the end of our 10:30, but I really, yeah, it is. But I really love this design. It's really, we'd love to actually have you join us. And I'll talk to you a little more off the record. In an upcoming series, we're interfacing with a few different biotech companies. A few of them are focused on APCs. So this would be really a great fit for you, I think, to contribute to the conversation. But I really, I think it's pretty amazing that this technology is developed to where it is, and I give you just a great amount of credit. You're a young scientist. You've really contributed a lot, and I'm sure in context of your mentors and everything else. But it really says a lot to you that you've gotten to this point. So. Congrats.
Well, thank you. I owe it all to all of the incredible scientists that I get to work with. I'm just surrounded by brilliant people. So they make me look good. It's not me, I promise. It's all them.
That's very humble. But anyway, well, thank you again, Jessica. This was just really great. I think Sarah left, but thank you again for joining us today. And I guess we'll leave it at that. I think I'm certain we're going to hear more from you in Intolerance. So I can't wait to see what you next.
Well, thanks so much. This was fun.